Hi, my name is Mariko Quinn, and I'm currently a rising sophomore at UH, majoring in global environmental science with a minor in political science. Even though my major is in environmental science, my primary interest is in marine biology, specifically coral reef ecology. For the last few years, I have been an intern at the Smithsonian Institution's Hagedorn Lab, located at Moku'olui, otherwise known as Coconut Island or the Hawaii Institute of Marine Biology. As an intern, I have been able to assist with a variety of different projects looking at coral reproduction, restoration, survival, and growth. However, the Hagedorn Lab's primary focus is on coral restoration through cryopreservation. Coral reefs are crucial to the health of the world's oceans and therefore crucial for the health of people around the world. However, as the health of coral reefs worldwide continues to decline as a result of a variety of stressors, it is becoming increasingly important that we have a variety of solutions prepared for the future. Cryopreservation is a relatively new method of restoration in which gametes, larvae, or embryos of a species are frozen in liquid nitrogen and kept in a state for potentially hundreds of years. In the event of a large die-off of a species, these samples could then be thawed and used for restoration purposes. Cryopreservation is a really important method of coral restoration as it maintains or even increases genetic diversity in a population. Many restoration methods specifically for coral focus on asexual reproduction and restoration, which could be problematic in the event of a large-scale die-off or large-scale restoration effort, as genetic diversity is crucial to the long-term survival of a population. Since genetic diversity is so important, cryopreservation is becoming an increasingly widely used process, and animals, plants from around the world are being cryopreserved. Since these samples are so important and so delicate, once an animal has cryopreserved gametes or anything like that, they are kept at incredibly secure facilities, such as the USDA facility in Colorado. These facilities are incredibly important and very well protected, as they house delicate genetic material that would be used in the event of a large-scale disaster or anything happening. Many of the species that are kept at these banks are also generally endangered, which makes them even more important. The Hagedorn Lab itself has successfully cryopreserved and banked gametes from 37 species of coral, and those samples are all kept at the USDA facility. While this is an incredible feat, another important aspect of coral restoration is the symbiotic algae. Zooxanthellae are single-celled algae that live within the coral host and provide it with its food. In the event of any type of stressor, they often expel these algae, which leads them to give that white appearance that is known as coral bleaching. This can often lead to death if the coral fails to uptake new zooxanthellae and get the nutrients that it needs. Therefore, symbionts are so important to these corals, and in order to really successfully restore corals, we need to think about the zooxanthellae as well. Therefore, for the last year and a half, I have been working with Dr. Baumeister to develop a protocol for the cryopreservation of coral symbionts. This is a very difficult protocol to develop, as symbionts have to go through a lengthy multi-step process to ensure that no ice crystals will form during the freezing process and to get the best survival from all of the samples. The first step is to extract the symbionts from the corals. To do so, we use a dental pick to strip the coral of its tissue, as you can see here. We primarily use this coral, mushroom coral, otherwise known as Lobactis scutaria, to extract the zooxanthellae from, as they are able to survive the process and can regrow their coral tissue, which other corals can't. Once the tissue is extracted, the samples go through a long filtration and spinning down process in order to clean and concentrate the samples as much as possible. The full extraction process takes upwards of 3 hours. Once a concentrated solution of zooxanthellae is developed, the samples are then encapsulated in alginate, which is a gel solution used to protect the samples from the highly intense freezing process ahead. In order to achieve successful freezing, the samples are spread incredibly thinly on a plastic blade, which is important for freezing later as well as thawing. After being successfully encapsulated, the samples then go through a series of different vitrification and equilibration solutions. Vitrification refers to the ability of a sample to be frozen without ice crystals. If ice crystals are formed, the samples will have died. Therefore, these solutions are needed to ensure highest survival rate. However, the chemicals in these solutions can be toxic to the samples themselves, so the process has to be done gradually, and that's where the equilibration solutions come in order to ensure that that process goes smoothly and delicately. Following that 2.5-hour equilibration and vitrification process, the samples are then covered in a final, highly concentrated vitrification solution and frozen immediately in liquid nitrogen. From here, the samples are stable and can stay in this state for potentially hundreds of years, or thawed by a laser when ready. With our samples that we freeze here, we keep them in a bank that we have here in our lab of liquid nitrogen. However, if we were to try to store these samples long term, we would ship them to the USDA facility for more safekeeping. Since this process is so lengthy and the samples are so delicate, much of the work that I have helped with over the last year has been to really refine the process and play with different concentrations of equilibration solutions and timings of different solutions in order to ensure the highest survival rate possible. Now that we have become more confident in this freezing protocol and we're starting to see really high survival and success rates of our samples, one of my projects for this summer has really been the next phase of this cryopreservation project. This phase is where we are trying to get coral larvae to uptake cryopreserved symbionts. This would really prove to us or show that this protocol is viable and working, as it would demonstrate that larvae are able to use cryopreserved symbionts just as well as they are the fresh symbionts. This is an incredibly exciting next phase that we hope to see success with by the end of the summer, if not within the next month or so. If and when we are successful in getting larvae to uptake our cryopreserved symbionts, we will know that this protocol is viable and can be used on a larger scale, long-term project, and hopefully for restoration worldwide. Thank you.